DNA, we have this long, long strand. And we don't want to deal with the whole long strand. We might just want a little snippet of it at that gene, okay? So what we need to do is we need to take the DNA from large to small, right? So here is your whole long sequence of DNA. It's much, much longer, but I'm just using it like this. And what we do is we take these things called restriction enzymes. They're defined as an enzyme that cuts DNA at a specific sequence of nucleotides. It's like a lock and key. It's looking for a specific sequence, and if it doesn't see it, it keeps going by. And once it sees a sequence, it's going to stop and cut it at that point. All right? It's like if you're searching for a file, and if I type in cell on my computer, it's going to throw every other file that doesn't have the word cell in it. And it's only going to give me the ones that I need. If I can be more specific, like cell extraction lab, it's going to look for something that's exactly that title. And eventually you can narrow it down and narrow it down. So we're going to cut DNA at a specific sequence of the gene. Okay, that's what the restriction enzymes are for. All right? We know hundreds of these. And then once we cut it, we need to analyze it. Now, this is a very, very complex technology. Here, I'll give you guys a minute. What's up?
Wait, so scientists cut the DNA?
Yeah, they have this chemical. The chemical is the restriction enzyme. So it takes the whole DNA sequence and it goes through, and it knows what code it's looking for. It might be looking for ATTAG, okay? And it's looking for that code, and unless it finds that, it's going to keep going. And once it finds it, it's going to cut that sequence, and it's just going to take out the little part. All right? You with me?
Yeah.
All right. All right. So you're going to separate the DNA now. So you may have had many different restriction enzymes. Let's say we were looking for a gene for hair color. We were also looking for the gene for eye color, and also, let's say, tongue rolling. Okay, I don't know. So you're going to take your DNA and you're going to add that restriction enzyme. Okay, and it's going to be in this little tube here. And this is called a gel. Okay, this procedure is called gel electrophoresis. Okay, we're going to model this next week in a murder mystery lab. It's fun. It's fun. You guys are going to be detectives. So you'll get to see the banding patterns that we're going to present from the crime scene. All right, so what happens is we take our DNA, we add our restriction enzymes, and we have this gel. And this gel is where we're going to load our DNA. There's these tiny, tiny little wells, and it's very, very hard to get this stuff in, because if you poke through the gel, then your experiment is pretty much screwed up. So you have to add all your chemicals, all your samples in, and then what you do is you're going to cover it with the solution, and you're going to hook it up to an electrical chamber. Now, DNA molecules have a negative charge. They're going to be put down at the negative end. And they're going to be pulled towards the positive end. Okay, they want to go towards the positive end because we don't want two charges right next to each other that are the same. We want them to go pulled toward the positive end. When that happens, larger fragments are going to go slower. Smaller fragments are going to go faster. So if you have a very, very small gene, that's going to get pulled up here, and it's going to travel farther. If you have a larger gene, it's going to not go as far. Okay. Now, let's say I'm going to pick on O'Toole. O'Toole.
Okay.
You were at somebody think of a store.
Giant.
Walmart. Okay. She was at Walmart. She's picking something up for Whiskers for the weekend. And what?
We had birds in it, and Whiskers was on that stuff there. Without abuse, I just saw that.
Was it the eagle? Yeah, I saw the eagle. There was 2 dead mice. All right. So anyway.
I'm so sorry. My homework's missing, and I think the guys might have mixed it up.
All right. Go in the back. Or and go there quickly. Okay. So, where was I? Okay, Megan was at Walmart. She was picking something up for Whiskers. And right before she left, there was a robbery. Okay. Megan was at the scene of the robbery. And the victim who was robbed said that it was a girl, maybe about 15, brown hair, suit, like, you know, her shoulders, and she was wearing an East Hills field hat or cross-country sweatshirt, okay? So they go and they say, you know, Megan O'Toole, we think that you robbed this place. And they take a sample of her DNA, because there was a sample of DNA at the robbery. They were really lucky that they got a hair sample left. So Megan's like, well, I didn't do anything. I didn't do anything. I wasn't there when I was, you know, this girl doesn't like me and, you know, all this other stuff. So what happens is they can prove that Megan was or was not there. And they can say, oh, well, you're lying that you said that you weren't there. Okay, Jackson, are you going to try to mess up my story?
I was going to say, what about cameras?
They didn't have any cameras at this Walmart. Okay.
Get out.
All right, so what happens is they look for a certain sequence of DNA, okay? And they see if that banding pattern is going to match up between the crime scene and Megan's. Now, Megan, you were lucky. It didn't match up. So they can conclude that Megan was most likely telling the truth. Okay? A lot of times, they will do this in crime scenes. They will get a piece of hair and they will get DNA evidence. And if their suspect can give a DNA sample, they will be able to match up whether or not that suspect was at the crime scene or not. If the blood matches his blood, they can pinpoint using this technology. It's very, very effective. So if the banding patterns match up for the two, and they're going to look for a specific enzyme, for a specific code, I should say, with those enzymes, they'll be able to say whether or not that person was at the crime scene, or if they killed the person, or if that person's the father of the baby, whatever. Okay? Oh, where did it go? Come back. Now, the next thing, okay, reading the sequence, okay? This just goes over the banding patterns again. And when you read the sequence, you need to have a couple different things. You need to have a fragment of a single strand of DNA. You need to have DNA polymerase, which we learned about during our DNA chapter. You need to supply the 4 nucleotide bases, adenine, guanine, thymine, and cytosine. And then you need each base labeled with fluorescent dye. Now, the fluorescent dye, you need to be very, very careful with. A lot of times you use something called ethidium bromide, and if you get it on your hands, it can be very, very carcinogenic. Okay? So you need to be, when I was working with it in college, I had to wear gloves all the time. We tried not to work with it a lot because it is dangerous. So, all right, now, the next thing, are you guys all finished? Is cloning. Cloning and transgenic. We're almost done, guys. I don't like to lecture this long, but I'm just going to go through these today. I have about 5 more minutes, maybe. Okay. An organism described as transgenic, all you need to write down is that it contains genes from other species. So we're taking, remember that tomato example with the elephant genes? Okay, to make it a larger tomato. That was a made-up example, but that's an example of a transgenic organism. You have a plant that is taking another organism's genes and has that in there, and we're using it for good. So all you need to write is that it contains genes from other species. How are transgenic organisms useful to human beings? We can produce a lot of different types of plants and animals that may have resistance to certain diseases or bacteria. Yes, Mario?
Do we write these with the little dashes on?
Yeah, give me 1 second. Now you can write these. There you go, okay? All right, so what about transgenic microorganisms? Transgenic bacteria produce important substances useful for health and industry. Transgenic bacteria have been used to produce insulin, growth hormone, and clotting factors. Now, insulin is extremely important for people with diabetes. If we can produce a bacteria that produces insulin, we may have a potential cure